course you've taken in this regard. Plus, it's not really that complicated. There's not a lot of probability, there's not a lot of crosses that you need to worry about. There's 2 units, right? So ABC was the first exam, C, part of it was on the second exam, D&E. So this one is F and G, right? And then we have one more exam which is coming up, and that is again independent of the other 3. So there's no cumulative exam. There will be cumulative things you have to know, but there's no cumulative exam per se. So any questions about at least the exam logistics? Same thing applies, right? You will be split into 2 rooms, same thing. 50 questions, 40 of them are going to be ABCD style, so true multiple choice, and then 10 will be true false. And you have the entire 90 minutes to finish it, so time should not be, in fact this should be the shortest of the 4 exams. I always start this with the request, if you are kind enough to donate your notes, please do. This, especially for this one, usually I have at least 1 TA present. They're going through major exams themselves, so they could not attend. So if you are in that boat, I know some of you have done it in the past, new blood is always welcome. You know, it's ultimately the nice thing to do, for what it's worth. Some of them actually are reading assignments. So, in fact, most of the chapters are reading assignments in the sense that I did not lecture per se. I lectured on chapter 11 because I think that's the most nuanced, especially when it comes to the Nassau-Stahl experiment. But the others are self-contained PowerPoints with a lot of concept checks and corresponding material in the textbooks and animations left and right for you to explore. So that's what I'm going to do. I'm going to go over them in order. We'll start with 9, and then we'll move on to 11, and then finally 12 and 13. 9 is stuff that frankly you should come into the course with. This is almost everything Gen Bio. So this is Bio 1, high school biology, however you want to term it. What you are expected to know and then be able to solve problems on are anything to do with DNA and RNA structure, right? So for example, and I know I've advocated, and then probably some of my other colleagues have done the same. I think, and again, take this for what it's worth, humble advice from someone who's been in the field for a long time. The more you write stuff, the more you remember things, right? So it's not enough to just like do note cards or listen to it. Actively involve yourself. So take a piece of paper and start writing what you know about DNA and RNA structure. So for example, it could take the form of a simple, start out with nucleic acids, right? Okay, so what do I know about nucleic acids? There's basically 2 types, right? There's DNA and there's RNA. Right? So let me break down DNA into 2 components, right? What are the building blocks of DNA? In fact, here's an example of a true-false question that you might get. You, just to show you that you need to know the stuff as a big picture and then detail. True or false? A nucleotide is smaller than a nucleoside. So a nucleotide is smaller than the corresponding nucleoside. True or false? Why false?
Because the nucleotide doesn't have a phosphate group?
Right, exactly. So you have to be able to know, things like that. So for example, what is the DNA, right? It's basically a polynucleotide, right? It's a polymer of nucleotides. So what is a nucleotide made up of? Actually, there are 3 components, right? There's the sugar, there's the base, and there's the phosphate. The sugar and the base make up what? A nucleoside, right? So how many sugars there are, right? There are purines and there are pyrimidines, right? So, and then the sugars, over the weekend I graded 120 essay exams, so I'm all over the place. I apologize. This is for the base. Let's just do this. Purines and pyrimidines. The purines are what in DNA, right? They're adenine, guanine. The pyrimidines are cytosine and thymine. The sugar is what? Is a pentose. right? What is it specifically called? Here's another question that while you don't have to know structure, you have in detail, you have to know overall. So which is bigger, the sugar in DNA or the sugar in RNA? Bigger in quotes, right? Which would be the sugar in RNA. Why? Because the DNA has one less what? Well, oxygen, right? It's called 2' deoxyribose, right? So you have to know that part right there. Right? Now again, you can probably, DNA versus RNA, right? What are the big differences? This is usually double-stranded, this is usually single-stranded. The RNA has uracil instead of thymine, and typically ribose instead of deoxyribose. I mean, again, you can do however way you want to flowchart it, but my point is that you need to be able to connect one to the other and be able to tell them apart, not just study them slide by slide. The RNA base is bigger because it's regular ribose. This one is 2' deoxyribose, so it has one less oxygen, right? Now again, you're not going to be responsible to know chemical structures, but you are expected to know relative sizes and relative differences. So for example, true or false, an adenine base is bigger than a thymine base, true or false?
True.
Why would that be true?
Because adenine is a purine.
Right, and then you have to know, remember, purines are 2 rings, right? These are 2-ring structures, pyrimidines are 1 ring. So while I will not give you like 3 double-ring structure and ask you which is adenine, I'll give you 3 structures. Only one of them is a double-ring structure. Which one of these is an adenine? That would be that one, right? Does that make sense? Now I'm giving you concepts now. I'm going to go over the problems shortly, because there's a lot of problems from that chapter. But any questions about concepts, right? You have to know basic nucleic acid chemistry too. In other words, what does antiparallel mean? One goes 5 to 3, the other one goes 3 to 5. What are hydrogen bonds, right? How many hydrogen bonds connect an adenine to a thymine? Connect, a guanine to a cytosine. And again, we'll address these points shortly. What is the only true or false? All DNA is right-handed. True or false?
False.
False, right? There is zDNA, which is left-handed. So you have to know that part. You have to know the diameter of the parameters of the DNA double helix, right? In fact, we'll work out problems like that. But any questions about the concepts in this chapter? Anything that you don't understand that I can elaborate on? Because then I'm going to go over some problems. And let's do some basic problems, right? We'll start with some and then go into higher ones. Okay? So let's say you isolate the DNA of a bacterium. And it turns out to have 15% adenine. So the adenine content is 15%. What is the content of guanine? How much guanine is there? And then here's the key. Let's do it real quick. So 15%, 25%, 35%, 45%, you can't tell. Now in these, and I've said it again, I'll say it again and again, can't tell means, I have no idea what you're saying, I can't tell. Can't tell means there's not enough information to tell, right? And in fact, can't tell is not an option, there should be enough information. And then the final answer would be what? If you've worked it out, the final answer should be 35%. Now how do you know that? Well, you have to know that the adenine always binds to what? Thymine. So if adenine is 15%, that means thymine is also 15%. So that means together they've occupied 30%. So what does that mean? That means G is equal to C is equal to what? 70, which is what's left, divided by 2, 35%. Questions about this style of... So let's take the same thing and then ask it to you in a different way, right? Same material. me ask you the same thing. And by the way, there's a lot of practice questions on Canvas or Connect, to help you along. So this is the base, and then percent. So adenine, cytosine, thymine, guanine, uracil. And let's say this is 10%. In fact, let me give you the key. Here's the key. Single-stranded DNA, single-stranded RNA, double-stranded DNA, double-stranded RNA, right? And then let's say cytosine is 20%, thymine 10%, guanine 60%, and uracil, of course, in this case would be 0%, because these have already added up to 100. Which is this molecule? It's a big enough molecule. It's a huge molecule. Which one would that be? Right? So it's not, it has 0 uracils and thymine, so it's automatically really not RNA. So you're left with single and double-stranded DNA. Now, which one is it? It's going to be single. Why? Because if it was double, A's and T's should be equal, which they are, but C's and G's should also be equal. They're not. So it's got to be single-stranded DNA. Does that make sense? So this is 1 or 2 points on the exam. It's simple stuff. C is not equal to G. A and T have to be equal, and C and G have to be equal. Okay, let me take the same information and increase a level of difficulty. So now we're talking about a single strand, okay? So here's the question. You are considering a double-stranded DNA molecule, and analysis of a single strand reveals that the A is 20%. So again, let me rephrase. You're considering a double-stranded DNA molecule, and analysis of one strand only shows that the DNA content of that strand is 20% adenine. What is the percent of adenine in the entire molecule? It's an open-ended question, right? This is a homework question, if you will. You have an unknown DNA molecule, and you're told that one strand has 20% adenine. What is the percent of adenine in the entire molecule? Exactly. This is a case where I need more information. I can't solve it. So then I go and I give you cytosine. Cytosine is also 20%. Is that enough information? So in fact, what information, let me rephrase it. What information would you need to solve this? Exactly, you would need to know what? You would need the thymine. The cytosine is not going to help you. You need the thymine, and here's why, right? If I go ahead and tell you guanine is also 20%, well, that's going to be enough, because if this, and this add up to 60, what's left is thymine, which should be what? 40%. Now, what is the answer? What is the adenine in the entire molecule, percent-wise? Can anyone figure it out? It's not 60%. If you use that logic, it's going to add up to 200%. It's not 60, it's not 40, it's what? There's 2 ways to do it, right? And then most of these, and I've said it again and again, whether it's in a lecture, online lecture, face-to-face, office hour, most of these questions, folks, you can answer in common sense, as long as you know basic biology, right? Basic biology tells me that if this is strand 1, right? If this is strand number 1, I'm going to have a strand number 2 facing it. So this is strand number 2. If there are 20% adenine, what's going to be the corresponding base here? Thymine, which would also be what? 20%. There's a cytosine here, while facing it would be a guanine, and it's also going to be 20%. A guanine here, facing it would be a 20% cytosine. and the thymine here, facing it would be an adenine, that's also 40%. Now what's the answer? It's not 60. It's 30, absolutely. It's 20% plus 40% divided by 2. That's your average. It's going to be 30%. If you use 60% as your logic, well, 60% are going to be adenine, right? Thymine is going to be also 60%. Now you're above 100%. It doesn't make sense. You have to average them out. So the answer is 30%. That's method 1. Method 2 is even more commonsensical, if you will. Here's method 2. And method 2 is I'm going to create a molecule that satisfies this condition. I'm going to create my own molecule that will have 20% adenine, etc., etc. Well, I'm not going to go with a huge molecule. I'm going to go with 10 bases, right? I'm going to go with a molecule of DNA that has 10 bases. So if 20% are adenine, that means I only have 2 A's, right? 2 out of that. I have 20% cytosines, that's 2 more C's. G's are also 2. And there are 4 T's, right? That's 40%. And then I'm going to create the complement, right? And I'm going to count how many A's there are. There's 1, 2, 3, 4, 5, 6. 6 out of how many? 20. That's 30%. That's a weird thing. 30%. Still. So if you have them on one strand and all of them, you're going to be able to tell the entire molecule. Questions about this style? Let's do a few more styles of questions on the concept. Let's do some sizes, I guess. So you isolate the DNA of a bacterium, and you are told it is it's 136 micrometers. So you isolate the DNA of a organism, and you know it's double-stranded DNA, and you're told that it is double-stranded. And the question is, how many base pairs is that? So this is a genome that is 136 micrometers long, end to end. So if you were to stretch it, becomes 136 micrometers. How many base pairs is that? So how do you solve such a problem? Right? I have to find a way to go from micrometers to base pairs. Right? What would be the value? What thing you've learned that will take you from a distance to a base pair? Sorry? Right. Basically, this is a DNA from a bacteria. 10 base pairs per turn. Now, a turn, I have a value for it, right? You should know that value. Not today necessarily, but certainly by Saturday. The value is 3.4 nanometers. That's your answer right there. 10 base pairs. This is actually something that you probably learned, the very first mathematical formulae you learned way back in middle school, kindergarten probably, right? Johnny has $10, okay, and an apple costs $2. How many apples can Johnny buy? That's exactly what you are doing here. I have every 10 base pairs gives me 3.4 nanometers. X is this, 136 micrometers. I need to solve for X, right? An apple costs $2. I have $10. How many apples I can buy? 10 base pairs is this. I have that. What is X? Right? First, I have to convert micrometers to nanometers. What is a micrometer in nanometer terms? And this is, again, the metric system. You should be familiar with it. A micrometer is 1,000 nanometers. A micro is 10 to the minus 6, a nano is 10 to the minus 9. So it's a factor of 10 to the third. So basically, times 10 to the 3, which will be equal to 136,000 nanometers. Right? And now I'm solving for x. Now whether you use a calculator or whether you use a, your hand or whatever it is, a mental, basically what is x? When you set it up this way, hopefully you remember, essentially what I'm saying is 10 over x is equal to 3.4 over 136,000 nanometers. So what is x? You cross multiply and you solve for x. So x is 136,000 times 10 divided by 3.4. That's solving for x. Now you can do it in whichever way you choose, but basically I'm going to take the point away and I'm going to add a 0. 136 divided by 34 is 4. So the answer is 4, 1, 2, 3, 4, 5. The answer is 400,000 base pairs. That's the answer. However you want to do it, the answer should be 400,000. Does that make sense? How many turns of the helix is that? It's 40,000. How did you get it? Just divide it by 10, because every 10 base pairs is one turn, right? So that's another style of question. Let me do a probability question based on the information. Let's do a probability one here. Now, while the exams are independent of each other, you still are required to know what's the end rule, what's the order, right? So this is basic, just like what is a base or whatever. You should know this information. So here's the setup. You're dealing with a DNA molecule where all bases are equal. So you have 25% adenine, cytosine 25%, thymine, etc. They're all 25%. And the sequence is random. So the sequence is entirely random. What is the probability that if I point to a sequence of DNA, I see this molecule or this sequence? Okay, 5 prime, let's say, A, G, R, A, N, T. Yeah, easy, huh? The sequence is grand. Now, G is a G, easy enough. R is any purine. Here's the given. R is any purine. And N is any nucleotide. So R is any purine, N is any nucleotide. What is the likelihood of finding a sequence that reads grand? So what is the likelihood, given the parameters that I discussed, G is found? What is the likelihood that you close your eyes, point to a DNA base, and it comes out to be a G? Sorry? 1/4. Why 1/4? There's 4 choices. I want one of them. So the G is 1 out of 4. What about the R? The R would be 1/2 because I have 2 choices that work for me, still out of 4. The 2 choices are adenine or guanine. These are your purines. So times 1/2 times also 1/4, easy enough. What about N? N is any nucleotide. So what's that probability? 1 out of 1. Everything works. It doesn't matter which nucleotide it is. So times 1 out of 1 times 1 out of 4, and use the AND rule. So this times this times this times that. Questions about this style? Yes?
Would it make a difference if they weren't all 25%?
Well, right, but then you calculate it with that probability,
Okay,
right? So then you would calculate each of the probabilities and then use that one.
okay.
So if A is 30%, use 0.3 as your probability, not 0.25. Okay, So one final question about, yes?
So do we use or if it says any order, like or when we use or?
Right. You would use the or, you would add them if I told you what's the likelihood to find this sequence or that sequence. So you calculate each probability and then add them up. Right, but for this one I'm not going to use. I'm just going to use the AND rule. I'm not going to use the binomial distribution or expansion now. Okay. The other thing you have to know, in fact, will be a good segue into the next one. Okay, so questions about 9. 11. 11 is probably, well, not probably, 11 is the single biggest chapter on this exam and it will garner the most attention question-wise. Right? So if you have time to study only one chapter, study chapter 11. 11 starts out with basic explanations of DNA replication. Now, DNA replication, there are 3 models for DNA replication. They could be the conservative, the semi-conservative, or the dispersive. You are expected to know what each is, and then how do you tell them apart. And this is where the Meselson and Stahl experiment starts with. So I'm going to spend some time on this one. You'll have 3 or 4 questions on this, and it's fairly straightforward. First of all, you do have to know that the method that actually is the method employed throughout evolutionary history is which one? The semi-conservative model. And the semi-conservative model, basically, imagine DNA is closed like this. The DNA opens up, and then you put a new strand here, and you put a new strand there, and that's your semi-conserved. Because I still have 2 molecules, but each molecule on its own is half old, which is represented by the arm, and then half new is the stuff that you put there. The other one is half old, half new. Now, what does conservative mean? Conservative, if you imagine, it's just like saying that your arms open up, you make a new DNA, but somehow your arms close back again, and that's your molecule, and the 2 created ones that are newly created, that is, recombine and make a new one. Okay? So that's conserved, because you have half old, half new. And dispersive is basically same idea. You still have to open them up, but when you create the molecules, each strand is part old, part new. So if you want to put it in color, okay, let's see if I have enough colors that work. If you want to put it in color, this is basically what we're looking at. Okay, so we are, and let's imagine our old is blue. Now, on the exam, you will not be tested on the steps of the Meselson and Stahl. You are not expected to know what is an isotope. You are not expected to know cesium chloride density. I'm going to set it up for you. But what I expect you to know is how to interpret the results. What do I look for if I have this versus that? So, remember, the key to this experiment is everything that is old is N15, which is the heavy DNA. And everything that's new is N14, which is the lighter isotope. It's light DNA. So heavy DNA is blue. Everything else is going to be red because it's light DNA. So let's assume the true method, which is the one that we know is true, which is semi-conservative. So what does semi-conservative mean? If you let this go for 1 generation, right, you're going to get what? You're going to get 2 molecules, half old and then half new. Does anyone have a problem with this? So if I take this and spin it in an ultracentrifuge, I'm going to get how many bands? Remember, this is the tube that you're going to spin it in. Now, this is where our normal DNA, if you will precipitate, will form a band. Where would you expect, which of these would you expect to be the N15? The one that is higher or the one that is lower? The N15, the lower, because it's heavier. So it's going to go to the bottom. So this is where you would expect N15 to go. This is where you would expect N14 to go. And if it's anything in the middle, it's going to be in the middle, right? So if you spin this construct or this solution, right, how many bands will you see? You're not going to see two bands because these are two molecules that are exactly identical. This is one molecule, this is another molecule. But they're both identical. So they will form only one band and then where? Right in the middle. So this will form a band right here. Okay? Now, take this and run it another generation. Right? So what do you think the banding, before the banding, what do you think the strands would look like? Which colors? If you want to recite them, or at least someone courageous enough. So first of all, how many molecules am I going to get here? Right? If I let it run around another round, I'm going to get 4 molecules. Right? What are the colors that I expect of each line, if you will? Right? I'm going to have what? I'm going to have blue, red, 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 blue. Why? Again, open, put 2 red lines. That's exactly what you're going to do. Open and put 2 red lines. That's basically what's going to happen. So I'm going to get blue, red, 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 blue. I'm going to spin this, right? How many bands will I get? I'm going to get 2 bands now. One where? One at N14, which are the red bands, and one is going to be in the middle still. Right? Let's run it for another round. Conceptualize this. I'm not going to draw them, but conceptualize this. If I run it for another round, right, how many bands will I see? If I run it for another round and then spin it, I'm still going to get 2, right, which are always going to be in the same position. But what's the difference? The intensity, which means which one of these is going to be thicker? The one at the top. Well, here it's represented as red, but it's the one on the top. So basically, after 3 rounds or whatever, you're still going to get this. The difference is that the band at N14 will get thicker. It's going to be more intense. That's semi-conservative. Right? Let's do conservative real quick. So this is semi-conservative, right? So let's do conservative. Conservative, let's keep the same code. Conservative, just like the term suggests, conservative means you've conserved and you're creating something new. So conserved, basically, after this is conservative replication. So if I spin it, I'm going to get 2 bands. I'm going to get one at N14 and then one at N15. Right? N14, N15. This is what I'm going to get. So I'm going to conservatively replicate this. Right? And again, I'm going to get 4 molecules, no breaking news here. What colors would you expect these 4 molecules to be in order from left to right? Right? I'm going to get blue, 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 red, 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 red. Why? This I'm going to conservatively replicate. So if I replicate this guy over here, I'm going to get blue, blue, and I'm going to get red, red. So now I'm going to replicate this one. Conservatively means I keep what I have, which is red. And everything new is also red. So then I'm going to spin, right? 14, 15. I'm still going to get the band at N15, right? The band at N14 will still be there, but what's the difference? It's going to be thicker. So let's conceptualize it, right? You do it conservatively, assuming it is conservatively. 10 generations later, right? What will the banding pattern look like 10 generations later? Conservatively, same exact with a minor difference, which is what? The 14 is thicker. On the exam, I can give you more intensity and more color, I'll give it to you as a flicker band. But it's not really that big, but the idea is that it's going to be bigger, right? Let's do conservatively, let's do dispersively. Dispersively is the hardest to draw, but in reality the easiest to visualize. So let's replicate it dispersively, right? Dispersively, same idea, right? Dispersively simply means that when I replicate my DNA, it's going to look something like this. I'm going to get part red and then part, the proportions are not going to be terribly accurate, but the idea is that each strand is part old, part new. So when I spin this one, right, how many bands will I get if I spin this concoction? One band. Where is it going to be? Right in the middle. Now it's harder to visualize, harder to draw it, right, for subsequent generations, but hopefully you can conceptualize it. If I do it just like we did here, I let it replicate, and then I spin them, right? After 2 rounds, how many bands will I see? Still one. But what's going to happen to it? It's going to go up the column because there's more red. So eventually, if you let it sit long enough, that band will keep getting higher and thicker. Eventually, the 15s will be diluted out, and you're really right at 14. Now let's do one numerically. Let's do one numerically, not visually. Numerically, without, again, the benefit of an answer. It's an open-ended question. Assuming conservative replication, right? Assuming conservative replication, you allow the bacterium to divide for 5 generations. Right? So I'm allowing it to divide for 5 generations. Open-ended question. What percent or what ratio of the molecules will consist of hybrid DNA? So assuming conservative replication, 5 generations later, what percent of the DNA molecules is expected to be hybrid? Hybrid meaning 14, 15 together. And the answer is 0. You're either going to get conserved or you're going to get light, heavy or light. Now, what is the ratio though? What percent or ratio of the resulting molecules is going to be heavy? Right? So 5 generations later, what percent or proportion of the daughter molecules at the end are expected to be heavy? No, the other way around. Right? 2 to the 5. So basically, figure out how many molecules do you have, right? 5 generations later. After 1 generation, how many do I have? 2, 4, 8, 16, 32. Out of these 32, how many are expected to be heavy? Out of these 32 molecules that I just counted, how many of them are expected to be heavy? 1. It's always going to be the one that's stuck at the end. 1 with 7 red. 1 with 15 red. 1 with 31 red. So the ratio is basically 1 over 32 or 1 to 31, whichever way you want to present it. But that's the idea. Right?
And it's not additive, so it's not like the 2 generations that we mix together in 1 year and add.
No, right. Because they're all dividing at the same time, right? Yeah, that's what we don't let it go longer because then they won't divide at the same time. Does anyone have a question about the Nussbaum stall? 3 to 4 points, so that's almost what? 7.5% of the exam. That's a big chunk, or 8%. You are expected to know the mechanics of DNA replication as well, knowing that it's semi-conservative. What are the enzymes, right? DNA polymerase. You have to be able to tell the difference between DNA polymerases in bacteria versus DNA polymerases in eukaryotes, right? In bacteria, we enable them with Roman numerals. DNA pol 1, DNA pol 2, DNA pol 3. right? Which is the major replicative enzyme? DNA pol 3. And again, you might not know it today, please know it by Saturday. In fact, ideally, know it by today. Which is the one that is only a repair enzyme? DNA pol 2. What does DNA pol 1 do? Right? It's important, but it's not terribly as important as the other one. Its role is to remove the primers. That's why it's required. In fact, it's called DNA pol 1 because it was the first one discovered, hence DNA pol 1. And you do have to know activity also, right? Which ones of these three? In fact, here's a key that you might get, and then several questions off it, right? And you know, that's my point in us, or at least advising you to do concept maps. Write the stuff down and see how it connects to each other. So you could do something like this, and again, using our frame of reference, right? DNA polymerases. There's 3 basic types in bacteria. And here we're talking about bacteria, right? We have DNA pol 1, DNA pol 2, DNA pol 3. Pol 1 is removal of primers. Removal of primers. Pol 2 is repair. This is the major workhorse. Now, here's the key, right? 5 to 3 polymerase activity. 3 to 5 polymerase activity. 5 to 3 exonuclease activity. And 3 to 5 exonuclease activity. So here are the key, right? Now remember, we make, well, let me ask you this. Which one of these have property A? Which of these 3 enzymes have property A? Which means they are making DNA 5 to 3. All of them. We make DNA, we make RNA 5 to 3. Right? All nucleic acids are created 5 to 3. So all of them are expected to have 5 to 3. Which ones are expected to have B? None of them, because that is against the canonical, against the norm. Right? Which one of these have 3 to 5 exonuclease activity? By the way, what is 3 to 5 exonuclease activity? Right, 3 to 5 meaning you're removing a base in the opposite direction. You're adding bases 5 to 3, so I'm going forward, if you will. 3 to 5 is I'm going backwards and removing a base. Why do I need to do that? That's proofreading. All of them have it. All of them have a 3' to 5' exonuclease activity. Which one is the only one that has 5 to 3 exo activity? DNA pol 1, and that's how it removes the primers, right? This is the only one. DNA pol 1 has it. C is only found in DNA pol 1, and that is the removal of the primer. So know that. That's as far as activity. You have to know also the, general features, right? In bacteria, they have a circular chromosome. When it gets replicated, what is that called? What is the mode of replication? Right? It's called the theta mode or theta because it looks like the Greek letter theta. So basically, when you have the chromosome opening up, so let's say this is the chromosome, and then it opens up. This looks like the Greek letter theta or theta, right? So, right, what is this construct called? Right, this opens up, this is called the I, but with the one that's going to the right is called what? Right, you have 2 replication forks. One goes clock, one goes counterclockwise, right? The part where DNA replication starts is called what? The origin of replication. True or false? The origin of replication of organisms tends to be rich in guanines and cytosines, true or false? Right? That is not that something that we discussed directly, but you have to be able to infer it, and the answer is false. Why? You expect it to be easier to open. G's and C's are harder to open because there's guanines and cytosines. G's and C's are triple bonded. A's and T's are double bonded. So they're less likely, G's and C's are less likely to open. A's and T's are much more likely. So do know that. You have to be able to put proteins in order. So for example, you have, in fact, let me give you an example. Because you will get several questions, whether it's transcription, translation, or replication. I'll give you several steps and ask you to put them in order, or several proteins and ask you to put them in order. So let me give you an example. Protein 1, and let's say is DNA pol 1. Protein 2 is DNA B. Protein 3 is DNA pol 3, and protein 4, let's say, is DNA ligase. I'm sorry, let's throw in 5 for good measure. DNA A. Right, so these are 5 proteins, and again, it's relatively early, you still have 4 days, but you should be able to answer questions like this, provided you think you know the material well. So, out of these 5, which is the first one that comes in, that lands on the origin, right? It's actually this one. That's the one that lands to the DNA A boxes. So out of these, the first one that lands is 5. 5 is followed by what? What is this enzyme? What does DNA B do? It's the helicase, which further opens up the DNA to allow replication to happen. So 5 is followed by 2, right, which is DNA B. And then does DNA pol 1, DNA pol 3, or ligase come next? Pol 3 comes next. Pol 3, followed by pol 1, followed by ligase. Now, why does pol 3 come first? Because that's the major replication enzyme. It starts replicating. DNA pol 1 comes later to remove what? The primers. And then ligase comes to seal the DNA with the last bit. So questions about this? Yes. Sorry?
What's the DNA exactly?
Well, A is actually the first one discovered, and DNA simply means the protein that binds to DNA.
That's the name of it, DNA.
Yeah, it's actually called DNA. And then we write DNA in small letters. So for example, primate is called DNA G. Also, and this is helicase. You have DNA C, which is the loader. We call them DNA. DNA stands for deoxyribonucleic, I mean simple DNA. And AB was the order in which they were, acting in this case. You have to know also the terminus, right? You have an origin, you have a terminus that's 180 degrees diametrically opposite, right? So we have 2 replication forks, one goes clock, one goes counterclock, and they meet at the terminus. And at the end, you have 2 semi-conservative replicated molecules, right? Now, what is the untangling of the chromosomes called? Right, that's the term that's also discussed. Sorry? Well, no, they're super cold when they are tangled. They have to be untangled, right? It's called decatenation, right? Catenane is Latin for chain, decatenation. That's the role of the DNA topoisomerase. Right? You are also expected to know the difference between leading and lagging, right? So what let me give you another key, and then that will hopefully ground your understanding. Because if you can't solve questions like this, frankly, I don't think you know it well enough. So A is leading strand only, lagging strand only, both strands, and I'll give you the questions, neither. So what I'm going to give you are proteins or events. Are they happening here only, they're happening both, or they have nothing to do with this, okay? So for example, if I mention DNA pol 3, right? DNA pol 3, does it work on the leading only, the lagging only, or both, or neither? Both. DNA pol 3 is the major replication enzyme. It's going to work on both. What about DNA pol 1? It's going to work on both, but what's the difference? Remember, what's the role of DNA pol 1? It's there to remove the primer. So there's going to be primers on both leading and lagging, but which one is going to have more primers? The lagging. So if the question was, the activity of DNA pol 1 is much more or higher on the leading versus the lagging, that would be false. But if it's required for both, absolutely. What about Okazaki fragment? That would be the lagging only, that would be C, right? What about Rho factor? That would be neither, right? Because the Rho factor is involved in what? Transcription termination, right? I don't have the notes from this chapter because I think that covers the majority. I think there was the end replication problem, and then we talked about telomeres. I think that was towards the end. Can I please look at the very end just to make sure I cover it? Yeah, you don't have to know the sequences of the DNA boxes, right? The consensus. Just know that they're rich with A's and T's. Elongation, we talked about that. Towards the end. Yeah, termination, DNA and telomere, and then I think you have to know the syndromes that I talk about. Yeah. Okay, right. You have to know what are telomeres, what function do they serve. And I think at the end we talked about progerias. Right. You have to know what are, progerias, the two types of progerias. We have adult progeria and, childhood progeria. So we talked about Werner syndrome, for example, and Hutchinson-Gilford. And you may have noticed, if you've listened to these lectures, I very humbly suggest movies and books, and Life of Sam is actually a very good short movie and documentary. He has YouTube. He has actually all sorts of stuff on the internet. He passed away recently, but he was the most famous, or one of the most famous progeria, child with progeria. In fact, his mom and dad founded a foundation to help discover a treatment for this disease, and they're actually well on their way to do that. But anyway, so I do that at the very end of that lecture. But you do have to know what are Hutchinson-Gilford versus Werner syndrome. What are telomeres? What do they serve? What functions do they serve? Any questions from 11? Okay, so 12 and 13 are related. They are relatively short, at least from as our discussion. So I'm going to quickly go through them. But same idea holds. You have to know the difference between transcription and translation. You have to know the polymerases involved, RNA polymerases, whether they're eukaryotes or prokaryotes. And at the very beginning, you do have to know the difference between template and non-template, coding and non-coding, etc. So let me give you a sequence, for example, and let's start our discussion with this. Let's say you have a C, T, A, G, let's put this as a G. So A, G, T, A, G, C, C, A, G. And the corresponding strand, of course, would be T, C, T, C, G, T, C. If my math, if my complements are all accurate. Okay, so here's a double-stranded DNA. And you're told that it's being transcribed. Okay? Which of the two strands is the template strand? So I'm giving you, or I've given you a double-stranded DNA sequence. It's a small part of a bigger region. And you're told that it's being transcribed. Which is the template strand? The bottom strand or the top strand? Which is the template strand? The template that is being transcribed. And bottom strand. Anyone say the top strand? Now the answer is you can't tell. It could be either. What do you need to know? What's conspicuously missing from here? Is ends. Okay, so I'm going to go and then give you ends and try to make you happy. 5 prime, 3 prime, 3 prime, 5 prime. I've given you ends. Which is the template? Bless you. Which is the template strand? Sorry? And you still can't tell because it could be either the bottom or the top. I still need one piece of information, which is what? No, the primers is DNA replication. Sorry? No, that's the protein, but it's related to that. You basically have to know where is RNA polymerase coming. Is it coming from right or is it coming from left? So the final piece of information is it's actually coming from the right. So RNA polymerase is heading this way. Right? So now which is the template strand? Sorry? It's the bottom one. Because remember, RNA polymerase is just like DNA, make RNA 5 to 3. So the bottom one is the template, right? So 3 prime will bind 5 prime, right? T comes off as what?
Would it be just the same as the current?
Well, I mean, let's get that. Let's get to that one. A T will be transcribed as what? As an adenine. A C will be transcribed as a guanine. An A will be transcribed as a uracil. A T as an adenine. A C as a guanine. Cytosine, cytosine, adenine, guanine, 3 prime. So notice this and this are exactly the same. The only difference is that you have a U here versus a T there. So what do we call the bottom strand? Template. What do we call the top strand? Non-template. But it's also called what? Coding, because it codes for the mRNA. It's exactly the same. You have to know mechanics. It binds. What is the sequence that the RNA polymerase binds to? No, again, that's translation. What's the sequence that is right before the gene that tells RNA polymerase where to start? It's called a promoter. RNA polymerase binds to it, and then it finishes at a sequence called the terminator. It either requires rho or it doesn't require rho. If it requires rho, it's called a rho dependent. If it doesn't, it's called rho independent. You have to know the difference between the core polymerase and the, right? You have the core, which is made up of what? Take that back. The holoenzyme, which is made up of the core, right, plus the sigma. Core and sigma make up the holoenzyme, right? And then in fact, there's alpha 2, beta, beta prime, sigma, and omega. It's in one of those slides, you have to know. Right? So true or false, the holoenzyme initiates, elongates, and terminates transcription. True or false? The holoenzyme, the big kahuna, initiates, elongates, and terminates transcription. True or false? False. How would you make it correct? Or why is it false? Because it's there at the beginning, but what is not there at the end? Sorry? The sigma factor. The sigma factor is not there at the end. So at the end you have the core and not the whole. So keep that in mind. These are again parts of transcription you have to know. At the end of this chapter, I go into details about the differences between transcription in pro and transcription in eukaryotes. You are not expected to know the details of the slides and the figures, that is. So notice in that part, I tell you you're not responsible for the details of this figure. So what are you responsible for? Basically, the big picture. So for example, which is shorter lived versus longer lived? Which is processed, which is not? Which has a division of labor? In prokaryotes, the same polymerase does everything. In eukaryotes, you have a division of labor. Messenger RNA is RNA pol 2, transcribe. What is a true or false? The mRNA in eukaryotes picks up a 5' poly A tail, true or false? Right? The mRNA in eukaryotes, when it's being processed, picks up a poly A tail at the 5' end. That's false. Picks up a 5' guanosine. 3' is the poly A tail. In the middle, what happens? The introns are, what's the technical term? Excised, right? Excision, surgically removed. The exons are spliced. Right? There's one form also that I discussed at the very end. It's called RNA editing. So it's processed at the beginning, 5', it's processed at the end, 3' in the middle, and it's also edited after the fact. And editing usually involves uracils. So keep that in mind. Right, questions about transcription. It's fairly straightforward. Translation, let's move on to translation. With translation, same idea. We start with a discussion of the genetic code. You do have to know the characteristics of the code, right? It's a triplet code, 3 bases code for a codon, okay? You have to know the start codons, or codon. You have to know the stop codon. You have to know what does true or false. A degenerate code refers to the concept where a single codon can represent more than one amino acid, true or false. So again, the degeneracy of the code refers to the fact that a single codon can give you more than one amino acid, true or false. False is the exact opposite. It's many codons giving you a single amino acid, right? And what are they called? If you have an amino acid, if you have, well, they're called redundant or degenerate. But if you have a codon that gives you an amino acid, what is it called? Sense. If it doesn't give you an amino acid, it's called a non-sense. Stop termination codon. How many? There's 3 of them. UAA, UAG, UGA. The start codon is almost always what? AUG, which encodes methionine, right?
Will you give us the table?
If it's beyond that, I'll give you the table, the code. If it's beyond those
Yeah.
start, stop, and methionine and tryptophan, the exception to degeneracy. Right. Beyond that, I will give you the code. Right. So you do have to also answer questions of this nature. Is the genetic code overlapping? That is, the genetic code is overlapping, true or false? False. Genes can overlap, true. It's a big difference, right? Let's do a math problem based on this, just to show you that probability can be used in almost every context. You isolate the DNA from an alien, and instead of the 4 classic bases, they have actually 5 of them. So they still have adenine, guanine, cytosine, thymine. So they still have these 5, but they have a new base, let's call it gozomine. Okay? So they have a new base, let's call it B. And they still have a triplet code. How many codons does their code have? Right? They have 5 bases, not 3, but they still use them 3 at a time. How many codons are represented in that code? What will be the answer? So if you have 5 and not 4, and you still use them 3 at a time, how many possibilities are there? And someone said, someone whispered, anyone? What is the normal code? Our code, let's say it normal. How many codons do we totally have? Right? We have 20 amino acids, but how many codons? We have 64. How did we get to 64? Sounds like someone, you know what, 64 sounds like a cool number. Let me plug it in. It's 4 to the third. That's how. It's the AND rule, guys. There's no magic. There's no miracle. It's the AND rule. Because this position can either be an A, a G, or a C, or a T. The next position can either be an A, a G, a C, or a T. The third position can either be this, or this, or that. So look at the different permutations you can have all over.